Okay, so I guess we can start. So I see we are already a few. So welcome everyone to this first webinar of the ISV NextGenVac for the year 2025. So I want to first wish you all a happy new year, and then let's go over with science. So first of all, today we will have two different talks. We will have Alexander Sampson from Oxford University, that was the Bright Spark winner of this year ISV Congress. And then we will have Dr. Manon Cox, that will that will present us some how to do entrepreneurship and biotech startups. So first of all, I would like, wait a second. I would like to remind you a little bit about ISV and why you should be part of it. So don't hesitate. The fact of being part of the ISV give you many different advantages, such as reduced registration for the ISV Annual Congress. Then you can have access to career mentorship for early career researchers. Of course, you can participate in ISV committees, but also if you're a young researcher, participate to the next generation vaccinologist group, and it's very fun. So I suggest you to come in. And then you have also other opportunities, such as the opportunity to network, which is always very good. And the fee is not expensive for one. If you're a postdoc or a PhD student, your one-year membership is only $35. So please join us. Also, I would like to remind you for next webinar of the ISV, which will be on the 22nd of January 2024. So don't forget to join. It's going to be a very, very nice talk. In case you want to be part and see a little bit how it works, so don't forget to join our channels. So we have a group on WhatsApp, so do not hesitate to join. It's going a little bit by itself. And also we have a LinkedIn page and a website, so go on to look to the newest updates. And if you have any question, don't hesitate to email us at nextgenvac@gmail.com. Also, one last reminder, so our next webinar will be on the 7th of February, and we will have two different speakers as usual. So James Allen from Cleveland Clinic and Michael Schotzert from the Ghent School of Medicine and Mount Sinai. So please write it on your agenda. It's going to be another very, very nice webinar. So now we can start with our first speaker. So Alexander, I let you share your screen, please. So Alexander Sampson, as I said before, he's the winner of the Bright Spark talk at the ISV conference of this year in Seoul. So he's a fourth-year PhD student at the Pandemic Science Institute, working with Professor Teresa Lambe and Professor Dame Sarah Gilbert. And Alex is originally a vet and developed an interest for emerging zoonotic pathogens with pandemic potential. So currently he's working on a next generation vaccine using the adenoviral vector platform that is famous in Oxford. So thank you very much, Alexander, to be part of our webinar, and the floor is yours.
Okay, so yeah, so hi, I'm Alex. I'm going to be speaking about trying to develop the next generation of viral vector vaccines using capsid display as a technique to try and improve the humoral immunogenicity of our vaccines. So as I mentioned, so I originally trained as a vet at the University of Cambridge, and then briefly worked as a research assistant on a vaccine, an early vaccine for COVID, before working as a vet for a year. And then I was sort of fortunate enough to get the opportunity to pursue a PhD with Professor Teresa Lambe and Professor Sarah Gilbert. So Tess and Sarah have been working for a number of years on a range of different vaccines against emerging pathogens, from Ebola virus, flu, Lassa fever, New World arenaviruses, and many others. But they're most famous for their work during the COVID-19 pandemic, where they developed the ChAdOx1-nCoV-19 vaccine, or the Oxford AstraZeneca vaccine. So this was a vaccine based on the ChAdOx1 viral vector platform, of which 3 billion doses were made during the pandemic. And it was responsible for saving around 6 million lives. So when we think about the viral vector platform, when you think about adenoviral vectors, the way they work is that they're a harmless common cold adenovirus that carries a foreign transgene from a different pathogen. And when they enter host cells, the only gene that gets expressed is this transgene, and we then get immune responses against what's ever encoded. So in the case of the Oxford vaccine, this was the COVID-19 spike protein. And traditionally, viral vectors have been associated with very, very strong T-cell responses. And we saw that again during the pandemic. So the T-cell responses, this here from this paper, showed that a single dose of the Johnson & Johnson adenoviral vector elicited stronger cytotoxic T-cell responses than 2 doses of an mRNA vaccine. But in contrast to newer platforms like mRNA vaccines, or also platforms like virus-like particles, the antibody responses that viral vectors elicit tend to be more modest. So this again is data from the pandemic, looking at responses to the SARS-2 receptor binding domain, a really important bit of the spike protein. And you can see that after 2 doses, we achieve about a log-fold higher responses using 2 doses of the Pfizer vaccine than we do with 2 doses of ChAdOx1-nCoV-19. So out of those other platforms, the one I'm particularly interested in is virus-like particles. So this is a platform, or there's a number of different platforms built using virus-like particles, where you have the repetitive display of the same antigen on the surface of a nanoparticle or an inactivated virus. So an example of this is the R21 vaccine against malaria that was developed at the Jenner Institute in Oxford. So here, the repetitive display of the NAN-P repeats, so that's repeats of 4 amino acids on the outside of a virus-like particle, elicits incredibly potent antibody responses. So the thought is that by repetitively displaying the same antigen in sort of very closely spaced, you can ligate multiple B-cell receptors in close proximity, leading to potent B-cell activation and therefore very, very strong antibody responses. So VLPs, these virus-like particles, elicit very, very strong antibody responses. But in contrast, like most protein-based vaccines, they tend to elicit fairly weak T-cell responses. So my PhD therefore set out to try to combine the strengths of both of these vaccine technologies into a single vaccine platform by displaying antigens on the outside of viral vector particles. So the idea being that using capsid display of antigens on the surface of the viral vector, we could still have an adenovirus that could enter cells and drive robust T-cell responses against the antigen that it encodes, but at the same time, we could drive potent VLP-like responses to antigens that we display on the outside. So in order to do this, I essentially had to initially identify proteins displayed on the outside of the ChAdOx1, or the more recent version called ChAdOx2, viral vector capsid that we could potentially modify. So there's several different proteins that are on the outside of adenoviruses. We've got the fiber protein, the penton, protein 9, or the hexon. And if we modified any of these to display a foreign epitope, that would then be presented on the surface of the adenoviral vector and could therefore work like a virus-like particle. Of these, the most obvious choice is the hexon protein. So this is the major capsid protein. There's 720 copies of the hexon in each virion. So if we genetically incorporate a foreign sequence into the hexon gene, that inserted sequence will be displayed 720 times on every single viral particle in the viral vector. So when we look at the structure of the hexon, they form trimers in the virus. And if we look at variability across different adenoviruses, we can see that it's almost all located, when we map that onto the structure, it's almost all located on the outside of the viral vector. And that's important for 3 reasons. Firstly, that variability has likely evolved in response to immune selection pressures. That shows us that if we inserted sequences into these surface-exposed sites, they would likely be exposed to the immune system, just as we would want from a VLP. At the same time, if we look at the protein itself, the variable regions tend to form unstructured protein loops. And that again is important, firstly, because these don't sort of form heavily structured regions, so they're unlikely to be sort of absolutely essential for assembling the virion architecture. So we could likely insert foreign sequences into these sites without disrupting assembly of the virions. And secondly, this variability means that they naturally accommodate differences in sequence and size. So we could likely remove regions that are sort of colored red here and replace these hypervariable regions with foreign epitopes that would then be displayed on the outside of the virion. So initially, to test this out, I had to identify different sites within ChAdOx1 or ChAdOx2 that we might be able to modify. So I took that key protective epitope for malaria, the NAN-P repeat epitope that's targeted by the currently licensed malaria vaccines, and incorporated that epitope into multiple different sites within the hexon gene. So something like this. So the NAN-P repeat epitope, it is literally repeats of the amino acids NANP repeated in tandem. So I took that sequence and incorporated it into different sites within the adenovirus hexon gene. I then tried to rescue the virus, and this took quite a long time to identify sites that were actually permissive. But eventually, I was able to identify sites where I could rescue equivalent titers of virus to the wild-type virus. And that's what you can see here. So on the left is a ChAdOx2 adenovirus encoding GFP, but displaying the NAN-P repeats on the outside of the virus. And on the right is just the wild-type ChAdOx2. And what you're seeing is this nice thick white band that we form after cesium chloride purification of the virus. So that's the virus compacted down into a single layer. And you can see that the titers that we were able to rescue from each of these was essentially identical. So initially, I was able to identify a number of different sites in both ChAdOx1 and ChAdOx2 where we could incorporate foreign sequences and still rescue equivalent yields of our virus to the wild-type vector. But I then wanted to check whether that actually corresponded to these vectors displaying the epitope to the immune system as we would want. So initially, I just looked at whether these sequences were incorporated into the purified virions that I could rescue. So on the left is just a total protein stain. So lanes 1 to 4 are modified vectors that are displaying this malaria epitope, and lane 5 is the wild type, just with the normal hexon. And the total protein stain looks the same, and you can see the band for the hexon protein at about 110 kilodaltons. But this is then a Western blot using a monoclonal that we know protects against malaria by targeting this NAN-P repeat epitope. And what you can see is that we get this nice staining at the same protein size as the hexon protein in all 4 of our modified vectors, but not in the wild-type control. So that confirms that the epitope is incorporated into the purified viruses. But I then wanted to check whether that was actually displayed and accessible on the outside of the virion. So I coated ELISA plates with these different viruses and then used the same monoclonal to probe whether we could actually bind to that epitope on the surface of intact viruses. And this is what we got as a result. So you see this lovely dose-responsive binding to all 5 modified vectors that are displaying the malaria epitope, but not to the wild-type control. So what that shows is that not only can we rescue viruses displaying these epitopes, but these epitopes are actually accessible in their sort of correct antigenic conformation to monoclonal antibodies that we know are protective against malaria when they're in the ChAdOx2 hexon. So having identified that we could display these epitopes, the next step was actually to check whether this was immunogenic. So we vaccinated mice either with, I had sort of 3 groups that were ChAdOx2 vectors encoding GFP, but displaying that epitope in various different sites within the hexon and different hypervariable regions. I also had a control that just encoded GFP, so just encoded the transgene, but had a wild-type hexon. And I also had a vector that encoded the full-length protein, the circumsporozoite protein, or CSP protein, that normally contains this NAN-P repeat epitope. So this would be the traditional way of making a viral vector vaccine against this particular antigen. So this vector encodes the full-length protein containing this epitope as a transgene. We then vaccinated mice with a prime and then a boost a month later, and we collected serum samples before boosting and when we culled the animals to check immunogenicity. So initially, if we just focus on responses to the NAN-P repeats, if we look at total IgG responses, after a single prime, responses are fairly similar across the board. So in the bluish color here, we have the full-length circumsporozoite protein, and the majority of animals seroconvert after a single dose. And using hexon display of the NAN-P repeats, we get similar responses depending on the site where we incorporate it. So the final group with site 3 seems to elicit the most consistent responses to the NAN-P repeats. But where things got really exciting was when we boosted with the same constructs. because the group where we vaccinated with a CSP control experienced a fairly mild boost, we saw a really substantial, almost 100-fold increase between prime and boost for all of our groups displaying the NAMP repeats on the outside. And the total, so the best group basically had a median anti-NAMP response up to 150 times greater by displaying the NAMP repeats on the surface of the adenovirus than we did with a vector encoding the full-length CSP as a transgene. At the same time, these vectors were still working to enter cells and carry a transgene, in this case encoding GFP. So we wanted to check whether responses to that transgene were maintained. And at both prime and boost, in all three of our modified vectors, we achieve equivalent anti-GFP responses to a wild-type vector that just encodes GFP. So overall, we can elicit up to 150-fold to surface displayed epitopes than we can achieve by encoding the same protein as a transgene. But at the same time, we are still able to deliver a transgene and drive an equivalent response against that as a traditional viral vector. So just to summarize where we're up to so far, we've identified sites that we can modify in both ChAdOx1 and ChAdOx2, these two different adenoviral vector serotypes. I've shown that linear epitopes that we directly incorporate into these sites can then be displayed and are accessible on the outside of the viral vector. And that can then drive really potent VLP-like antibody responses to those displayed epitopes while maintaining the ability of the viral vector to work in its traditional way in entering cells and driving responses against an encoded transgene. So this was very exciting, but I think there's a limitation, which is that most of the time, we're not interested in driving responses solely against a single linear epitope, just a string of amino acids. Normally, what we want to do is drive responses against native epitopes in conformational proteins. So this is where we partnered with a startup company in Oxford called Spy Biotech. So Spy Biotech have developed, they've sort of pioneered this work in human adenovirus 5, and they've developed a suite of technologies called SpyTag and SpyCatcher, which they also describe as like a molecular superglue. So the way this works is that the tag is a small peptide, and the catcher is a corresponding protein that will bind to the tag and then irreversibly covalently fuse. And that's a spontaneous reaction that when you mix the two together, the peptide and the catcher domain just spontaneously form a covalent bond with each other. So they've used this in a number of different VLP platforms to attach antigens on the outside of a virus-like particle. So something like this, where you could have a virus-like particle platform that's sort of produced displaying the spy catcher on the outside. You could then mix that with your antigen, with the tag domain, and then just upon spontaneous mixing, they will irreversibly couple to each other with a covalent bond. So we then set out to use a version of this called the dog tag and dog catcher to see if we could attach whole protein antigens to the surface of the viral vector, looking something like this. So the dog tag is a version that works well in loops, so we can incorporate the dog tag, shown here in yellow, into the hexon hypervariable regions. And the thought is that we could then incubate that with protein antigens like the SARS2 receptor binding domain, or RBD, fused to the dog catcher. And by mixing those together, you should then get that coupling reaction between the dog catcher and the dog tag, allowing you to display that antigen on the outside of each viral particle. So this is just a close-up of what this might look like. So the dog tag, which is genetically incorporated into the hexon shown here in yellow, forming this irreversible covalent bond with a corresponding dog catcher, which you could then fuse to something like the SARS2 receptor binding domain. And the idea being is that you could then form this array of an antigen on the surface of the viral vector. So I was able to rescue a number of different viruses, so ChAdOx2 viruses, with the dog tag incorporated into different sites on the surface of the virus. So I then incubated that with the dog catcher fused to the SARS2 receptor binding domain. And again, this is just a total protein stain that you're looking at. And this band at 110 kilodaltons is the hexon on its own. I then incubated this with an increasing concentration of the dog catcher fused to the RBD. And what you see is that as we increase the amount of ligand, we get the formation of this second, much bigger band at about 160 kilodaltons. And what that represents is now the hexon irreversibly covalently coupled to the receptor binding domain from SARS2. And by estimating the proportion of hexons in their sort of native state compared to the coupled state, we can estimate the proportion of the surface of each viral particle that would actually be decorated with these SARS-2 RBDs. And depending on the site, we could reach about 80% of hexon monomers being irreversibly fused to the SARS-2 receptor binding domain. So on every single viral particle that's delivered in the vaccine, you would have about 500 copies of the SARS-2 RBD displayed around the surface in a very dense array. So I was then really excited to see whether that was immunogenic, again in mice. So here we had 4 groups, each of which were, again, encoding a relevant GFP transgene, and on the outside, we're displaying the SARS-2 receptor binding domain. We had a control just encoding GFP, but with a wild-type hexon. And we had another group encoding the full-length spike transgene. So that's exactly the same transgene that was used in the Oxford AstraZeneca vaccine used during the pandemic. And we took serum samples just before boosting at 4 weeks and again after culling to look at antibody responses to the SARS-2 spike and at responses to the GFP transgene. So here, even after a prime, in groups 3 to 6 that are displaying the RBD, we can see that we're trending towards higher total IgG responses to the RBD than a group using a spike transgene. And again, when we boosted with the same constructs, that gap widened. So in all cases, we reach at least 10-fold higher median responses to the SARS-2 RBD when we use a vector displaying the RBD compared to when we use a vector encoding the full-length spike transgene. And these aren't just binding responses as well. This corresponds to functional neutralization as well. So using a lentiviral pseudotype assay, we see between 5 and 17-fold better neutralization of SARS-2 when we display the RBD on the viral vector surface compared to when we actually encode the full-length spike. At the same time, these vectors were carrying a GFP transgene. And if we look at T-cell responses to GFP, in all 4 of our groups that are decorated with the RBD, we see equivalent strong T-cell responses to the GFP transgene as we do in a wild-type vector encoding GFP. So depending on the site where we sort of attach the RBD, we can elicit up to 14-fold times better binding and neutralizing antibodies while maintaining the responses to the encoded transgene. So overall, just to summarize, what this work has shown is that I've identified sites where we can insert either linear epitopes or display whole protein antigens. We can then drive potent VLP-like antibody responses to the displayed epitopes while maintaining the ability of the viral vector to work in its traditional function and elicit robust T-cell responses against whatever transgene are encoded on the inside. So there's a number of different sort of pathogens where this might be really useful, from malaria, flu, or COVID, where we might want T-cell responses against one protein. We might want to drive neutralizing responses against either multiple antigens or particular epitopes on the surface of the viral vector. So this modified platform should hopefully provide a really exciting approach when we might be able to achieve that. So I'd like to thank my supervisors, Tess and Sarah, as well as everyone else in the Lamb and Gilbert group, as well as our collaborators at Spy Biotech. And I'd like to open up to any questions.
Thank you very much, Alex. This was a great talk. So for all the attendees, please, if you have any question for Alexander, write it in the Q&A session so that I can get them to him right now. So while we wait for people to write something, I do have a question for you first. So one of the problems, if I remember correctly, of the adenoviral vector platform was reactogenicity, especially in some groups of populations. Do you think adding these VLP epitopes will increase the reactogenicity even more, or you did not experience this? How do you think this will impact this?
It's a good question. I think it's something we can't fully address until this is sort of used in the clinic. What I would say is that part of the immunogenicity is going to be down to the reactogenicity is going to be down to the inherent properties of the adenovirus. And while I don't think that adding these epitopes to the outside will decrease that in itself, if this was dose sparing and allowed us to achieve the same immune responses with half or a tenth of the dose, then potentially that could lessen the reactogenicity that we might see if we were able to use lower amounts.
Thank you very much. So we have a few questions. So here I have Kevin that is asking, is it possible to directly incorporate larger peptides on the exon?
Yeah, it's another really good question. So There probably is an inherent limit to the size of what you can incorporate into the hexon itself. So you couldn't just directly incorporate the SARS-2 receptor binding domain, and that's partly just the physical nature of the steric constraints of what you can fit in. I guess the other thing to bear in mind is that a lot of the antigens that we're interested in would naturally go through the secretory pathways. The receptor binding domain for SARS-2, for example, only really folds in the secretory pathway and forms the correct disulfide and glycosylation that you'd see in the secretory pathway, whereas the adenovirus and the hexon would normally assemble in the cytoplasm. So you wouldn't normally get that in an adenovirus. So that's where sort of the advantage of using this dog tag, dog catcher coupling to produce something that's secreted in its normal sort of normal pathway, and then post-translationally attach that to the adenovirus later on, sort of has advantages there. So both from the size and from the complexity of the antigens you can attach.
Thank you. Then we have Claudia Lindemann that is asking, so first of all, she's thank you for your great talk. And how immunogenic are the exons and how high is the average coupling efficiency?
So in terms of the hexons themselves, or whether they're immunogenic, that's the main site that we see neutralizing antibodies against the adenovirus. So we know that we do drive responses against the wild type hexon. So that's partly the why it's useful to incorporate foreign epitopes into this site, is that we know that it's a site that the immune system normally targets. The other thing was in terms of for directly genetically incorporated epitope like the NAMPI repeats, that's going to be present in 100% of the hexons on the surface of the adenovirus. When we're using the dog tag and dog catcher, it depends on the site and depends on the concentration of the ligand that we use, but we can reach about 80% coupling, depending on exactly which site and yeah. But I guess that's something that also is likely to change depending on which ligand that we use. So if something is much bigger or much smaller, that might affect the percentage that we could couple.
Thanks. Then we have a question from Manon. Sorry, you have a lot of questions, but that's super good. So Manon is asking if the integration site is specific for the antigen being studied, or it can be used for all antigens.
It's a really, really good question. I would love for there to be a single site that's perfect for all of them. In our hands, there's a couple of sites that seem to be particularly promising for all the different things that we've tried. But it is definitely possible that there could be some that would tolerate certain conformational antigens that others wouldn't. So it is possible that there are some sites that would tolerate a particular epitope, whereas another one wouldn't. So it's potentially something that you would have to try out for each de novo time that you used it. Whereas using a transgene in the traditional adenovirus way, that's fairly confident that sort of we tend to see very good immunogenicity regardless of the transgene.
So then we have, unfortunately, we have a lot of questions. So I don't know if you want to share your email or anything. So
yeah,
I will still take two of them, the next two. And then for the rest, if you have this question, you can address the email address of Alexander so we can continue with the next part of the session. So we have Eddie Sayed that was already saying great work. And have you looked at the genetic stability of this construct? Have you looked at durability of the immune response?
So genetic stability of the construct in the adenovirus, yes. So that's something that we're looking at currently, but it tends to be genetically, or it has been genetically stable for a number of passages, definitely enough for us to purify it. We've not seen any, even with larger and more complex inserts, we've not seen any sort of recombination events or shortening or loss of the insert. In terms of the durability of the immune response, that's something that's ongoing. So it takes a long time to see how long something lasts for.
Perfect. Then we have still last questions I will address. So Egan Jacobus, so need talk. What is the coding capacity of the site in the exon, and does the decoration of the adenovirus abrogate anti-exon responses?
Good. That's some really good questions. So the first one, it's something that again we're looking into. It likely depends on the site itself, and potentially also the epitope itself. So there's something that might be tolerated that in one site of a certain size that wouldn't be tolerated in another site. In terms of whether it reduces response to the hexon itself, what we do see is a partial escape from pre-existing anti-vector immunity. So we've seen basically if we decorate with the RBD on the outside or just genetically incorporate foreign epitopes, it does look like that changes the antigenicity of the vector, and so we see reduced recognition by serum raised against the wild type hexon. So it looks like that we do see some not total but partial escape from anti-vector immunity.
Okay, perfect. Then thank you very much, Alexander. This was a great talk. So let's see if we have some time at the end. There's still a few questions. I will get to you again. So now I would like to pass to our next speaker. So Dr. Manon Cox, welcome. So you can share your screen. We are very happy to have you here. So Manon Cox, Dr. Manon Cox is at the moment on the board of the ISV and is going to be the incoming president. So congratulations. And she's going to talk to us about entrepreneurship and biotech and startups. So please, the floor is yours.
Okay, Alex, great talk, and I'm happy to, we're going to shift gears a little bit from deep science into more of entrepreneurship, biotech startups, and just give you a view on what one could do after you finished your PhD and you are looking for other opportunities. So I want to start with my background in a nutshell. I'm a molecular biologist by training. I studied in the Netherlands at Nijmegen University. After that, I spent about 12 years at Hippocrates, where I was involved in the development of Lipomax, and I will speak a little bit about that. I have my doctorate from the University of Wageningen, also in the Netherlands, and I have an MBA from the Simon School, Rochester, United States, and that was actually how I ended up being located in the United States and getting involved with the development of Flublok, where I spent basically 20 years on getting the first recombinant influenza vaccine approved by the FDA. Over that time, we received about $100 million in support from the US government for pandemic preparedness. And turned out that this Flublok was such a great vaccine that Sanofi acquired the company. And because I was CEO at that time, they really felt that there was no need for me to continue to be involved in the company. Today, I have a couple of board memberships, and boards are organizations that basically help with the strategic direction of companies. One of those companies is Vaccess, located in Australia. GPN Vaccines, also located in Australia. I'm a former trustee of the University of St. Joseph in Connecticut, and I'm on the board of the Netherlands America Foundation to also honor my Dutch heritage, and on the board, as mentioned earlier, of the International Society of Vaccines. More recently, I've edited a methods book for Nature Springer, and I invite everyone who's interested in working with the baculovirus system to take a look at that book, because 25 great articles on how to work with insect cells are included in that edition. And I provide a lot of support and advice to multiple biotech and vaccine startups. So that's in a nutshell. What I would like to do today is I want to talk briefly about the development of this detergent enzyme, lipase, that I was involved in for about 10 years. I want to speak about Flublok, the development of the first and only FDA-approved recombinant influenza vaccine. And then after I left protein sciences, where I spent about 20 years really focusing on this one thing, I really had to answer the question, like, what am I going to do next? And because I came to the conclusion that I might not really be an entrepreneur, I decided to give you 3 real-life entrepreneur examples. The first one is Alina Venerio, who founded the company V-Vector Bio, Byron Martini, the founder of Artemis Bioservices in the Netherlands, and Peter Leonardi, the founder and CEO of Omnisite. And then I'll share some take-home messages. So if I look back at my career, I can actually say that I was really always focused on getting something done from beginning to end. So Lipomax is actually, I was involved from the idea to the ultimate product that ended up in detergent enzymes. So if you buy a detergent from Unilever or Procter & Gamble and you have greasy stains in your clothes, in principle, Lipomax should help remove those stains. So you can imagine that conditions in a washing machine are very problematic for multiple enzymes because there's a super high pH and, we wash often at higher temperatures. So Hippocrates was a large biotech company in the Netherlands. They had a whole department that would go out in the world to screen for organisms that would produce those enzymes that we needed for various applications. And in this case, they found a Pseudomonas alkalicinus lipase that actually was able to produce a protein that had a very high pH optimum. We already used, and this is now probably around 40 years ago, we already used protein engineering to modify certain cysteines in the protein sequence to produce an oxidation-resistant version of this product. And recombinant DNA technology and classical mutagenesis was used to improve production levels to a level from a few micrograms per liter to 12 grams per liter, making this product, basically viable as a commercial entity. And I was involved then after that with the process where the production facility was built for this enzyme, the purification facility, and I spent some time in manufacturing where the facility was built, running a process development group, which was a real exciting experience. But as a result of this program, I was recognized by the company to enter into a management development program that enabled me to do the MBA in the United States. And after that, I decided that I wanted to go back to my roots and really work on a pharmaceutical product. So I joined a company called Protein Sciences. Protein Sciences was a company of about 30 people. We were working on all kinds of vaccines. And what we did at Protein Sciences, we basically developed the recombinant influenza vaccine. I speak a little bit more about that because Flublok is a very interesting product. It has, unlike other influenza vaccines, doesn't require the use of eggs to make the vaccine. It has a higher antigen concentration. We were not dependent on growing a live influenza virus. We didn't need preservatives in the vaccine, and we also didn't use antibiotics in the production process. So the baculovirus production system enables you to actually take advantage of all the characteristics of a plasmid and bacterial production, and yet you can use this virus to infect in a transient manner insect cells and produce your product of interest. So we were able to basically develop a process applicable to all the influenza viruses because you may not be aware of the fact, but influenza vaccines change every year. So in other words, you needed a production process that was very rapid and that was able to deal with those rapid changes. And we demonstrated convincingly that we could go from gene to production in 22 days by using, of course, PCR, direct cloning, creating a baculovirus expression vector, and then developing a universal purification process that was just based on fermentation, harvesting by centrifugation, extraction of your protein of interest, clarification, capture of the protein, purification further, removing the DNA to an acceptable level, and then using tangential flow filtration to come up with the final formulation of the product. So during this 20 years at Protein Sciences, we spent a lot of time doing clinical studies in which we demonstrated that Flublok was able to prevent you from getting the flu. And it actually did that in a way that was superior to the licensed egg-based vaccines. So while the development of Flublok probably took 20 years, it was really a very intensive process even after product approval, because then we really our initial approval was in 18 to 49 years old, and then we had to expand to actually the population that really requires or benefits from a better influenza vaccine. So we needed to do a lot of additional post-licensure studies. We needed to get licensures of production facilities that could produce larger quantities of product, and that required a multitude of clinical studies that ultimately resulted in the full licensure of the product for people 18 years and older. And the ability to manufacture this product at a 20,000 liter scale with help of a Japanese company. So it was really great to see an announcement in 2019 or a publication in 2021 from the FDA that basically showed that the recombinant influenza vaccine, Flublok, was significantly more effective in preventing influenza hospital encounters than all other vaccine types, except for the adjuvanted influenza vaccine produced by Seqirus. The recombinant influenza vaccine was also significantly more effective than all other vaccine types, including the adjuvanted vaccines, in the inpatient stays outcome analysis. So that was really, a very nice crown on the work. But it also resulted actually in 2017 or early 2018, the acquisition of Protein Sciences, which had by that time grown from a 30-people company to a 150-people company. And it was at that moment in time acquired by Sanofi. And Sanofi has done an amazing job in making the product far broader available, because we weren't able to sell more than 300,000 doses per year. And Sanofi is at this moment in time selling close to 10 million doses per year. So the good news about that acquisition was that the product is now far broader available, which is fantastic. But the bad news was that I was no longer associated with the company, and I had to think about what to do next. So I initially thought that I was going to start a company, and I realized that that I didn't really have the passion and the energy anymore to start all over again and to spend another 20 years to make something happen. So what I decided to do is to focus on board positions, helping companies with, for example, in the case of Vaxis, with the needle-free future of vaccination. And it's really exciting technology where hopefully in the future we will be able to self-administer our vaccines, have more stable vaccines, have better immunogenicity. I also joined the board of GPN Vaccines, which is trying to develop a universal pneumococcal vaccine. Really exciting, because for those of you familiar with pneumococcal vaccines, we start adding more and more serotypes to the vaccine. And as we do that, we basically displace the circulating pneumococcal bacteria with the ones that are not in the vaccine. So it's almost a race that you cannot win. And a universal pneumococcal vaccine would actually allow you to deal with that problem. I mentioned the Netherlands America Foundation. This is an organization where we actually sponsor people from the Netherlands to go study in the United States, and people from the United States to go study in the Netherlands. And we have also broad cultural programming in that organization. I'm a member of the board of ISV, and as mentioned, I will be the incoming president during 26 and 27, and I edited a book for Nature and Springer. I also did some very other different things. I was an executive producer on a movie about the comeback of a fighter, a boxer, and it probably will soon be viewable on Netflix and all those channels. And I'm a scientific on a number of scientific advisory boards, amongst others, Epivax, Therapeutics, Omnicide, Vivector Bio, Artemis Bioservices, and Intravec. back. And in those situations, I work with real entrepreneurs, people that are, really driving innovation in their field of interest. And I asked 3 of them to just give us some insights in how they view their role as an entrepreneur, what is great about it and what is not so great about it. And I will start with Alina Venerio, who founded the company VVectorBio, and she's developing the next generation of technologies to transform manufacturing of viruses for therapy and vaccine applications. And I know Alina as a person who's extremely driven and really, dealing from day to day with the challenges of being an entrepreneur. So she really wanted to start this company to use her expertise in virus manufacturing, and she really wanted to take control of this and have the freedom to create impactful technologies for patients. She is driven by an entrepreneurial spirit and passion for innovation, and you can see and feel that when you work with her. So it inspires her to work with passion, unlike any other job before. The adrenaline of constantly, risk keeps me motivated. Remember, as an entrepreneur, you are responsible for the people that you work with. You need to make sure that you make payroll. There's challenges, of course. And it has led to immense personal growth and new skills beyond the lab, like communication and management. And as her greatest challenges, she describes that gaining the trust from companies that she wants to work with and proving that you can do it is a major challenge. And commercialization of a technology requires effort, dedication, multiple strategies, and you have to stay confident no matter the circumstances. So I am confident that Alina will get there, and she's doing a great job. So Byron Martina is the founder and CEO of a company in the Netherlands called Artemis Bioservices. And Artemis Bioservices has been around for a while. What they're doing is they're assisting biotech and pharmaceutical companies in accelerating the development and licensing of safe and effective intervention strategies against viral diseases by providing customized in vitro and in vivo immunogenicity and efficacy testing. And when I say, you need persistence, we can indeed think about Byron, because his company has already gone through a multitude of, alterations and adjustments on how can we do best. And what Byron says, what he loves about being an entrepreneur is it allows him to turn ideas into impactful solutions, collaborate with brilliant minds, and drive innovation that directly improves global health. And he thrives on the opportunity to push boundaries and reinvent approaches in viral immunology. What he views as his greatest challenge is balancing the innovation with the practical realities of running a business. Securing the funding, and I mean, this is big, as I know from my personal experience, navigating regulatory hurdles, building trust, same thing as what Alina says. And he does say, these challenges fuel my determination to succeed. His final quote is from passion to purpose. So he really leverages his expertise that he basically acquired in academia to drive innovative solutions and accelerate life-saving breakthroughs. He founded Artemis to bridge the gap between research and real-world solutions, and turning ideas into regulatory compliant products is challenging, but it drives our mission to support the biotech industry and deliver breakthroughs that save lives. And again, I see Byron as somebody who is just at it and doesn't give up. And I think that that's really what you need to, to be successful as an entrepreneur. And then finally, Omnisite. Omnisite is really a vaccine development company that is trying to find targeted life-saving vaccines for patients with cancer, diabetes, and multiple sclerosis, amongst other things. And what Peter says is, I have a unique background, both academic, where I studied at diverse institutions, and also the US Air Force Academy, New York Medical School, and Carnegie Mellon Business School. In business, he worked at emerging companies such as Protein Sciences. That's where I obviously know Peter from, and he helped us there in the financial department because he had just finished his MBA at that moment in time. But he also worked in large companies like IBM and New York Presbyterian. He feels that what made him an entrepreneur was his diverse set of experiences. He believes it's his stubbornness and his unwillingness to accept the status quo that really makes me an entrepreneur. And when you have to be resilient to continue, you have to be resilient to continue when you hear and see negative results over and over, and many tell you it can't be done. It's just that one big positive result that makes it all worthwhile. So Peter, again, is never giving up. He's focusing most of his efforts in trying to raise money for the organization by writing grant applications, by meeting with investors. And again, it's about never giving up. So if I think about entrepreneurship, then you can ask yourself, well, what does it all mean? I think you have to have a ton of passion for what you're doing. You really have to believe in that you can make it happen. Persistence, resilience, you need a lot of energy and drive because often things will not go the way you think it should be going. And of course, a lot of you who are working on a PhD, you see that as well, right? I personally think that focus and planning and having a great team around you is super important because you can't do it all by yourself. And it's about never giving up. So with that, I want to also make a little bit of a plug of a mini symposium that I've been able to put together with Mark K. Hobbs from Pfizer. And in this mini symposium, we're going to deal with pneumococcal vaccines, and it will take place on January the 30th, and it starts at 9 o'clock and it should be finished by 11 o'clock. And we're going to have some great speakers. We're going to have the president of the International Society of Pneumonia and Pneumococcal Diseases. She will speak about what they do. We have a former FDA regulator who is now on the board of the chairman of the board of the Sabin industry, a speaker from Pfizer, who'll give a general overview on the impact of vaccines on pneumococcal disease. And we have some real life impact data following switch of vaccines from a speaker from Belgium as well. So with that, I would like to thank you for your attention. And if you have any questions, email me. And as Benjamin Franklin stated, energy and persistence conquers all. So with that, I want to thank everybody for your attention, and I'm happy to take questions.
Thank you very much, Manon. That was an impressive presentation. So I would ask already to the attendees, if you have any question for Manon, please write it in the Q&A. And in the meantime, I will start. I have more a question that is, it's something that I was wondering since a long time. So I've seen you, you said you did an MBA at one point, and how much was that actually useful for your future career? So was that something that was requested to you, or did you choose to take it spontaneously? Would you recommend it, or?
Yeah. Well, I think it was an amazing experience because when you are being trained as a scientist and when you do your PhD, you go very deep into a relatively small topic and subject. And at Hippocrates, the HR program was actually such that they really wanted to have young people of whom they thought, well, they can be important for the company. They wanted them to get a broad experience, which is why I worked in manufacturing. I worked 9 months. I worked 9 months as an assistant to one of the top managers, and I worked 9 months in business development. And they sponsored me to do an MBA. And this was like a small group of people that they had identified. And the idea was that you would really learn more about what it takes to be an entrepreneur. And what my personal experience was, it was just amazing to, in a 2-year executive program, to learn so much on such a broad range of topics. And I would basically say that it was very important to do that, because it allowed me to broaden myself and to just become a different convincer of people, right? As a scientist, you always rely on, like, this publication says this and what have you. Whereas in reality, if you want to raise money, it's more about exuberating confidence and showing that you know what your mission is and where you're going, and etc. And that's what I learned in a short time. And what I feel, and I see that there's a question, I feel that it makes most sense to do an MBA after you have already obtained some experience in the industry. Because if you do it right out of school or right out of coming out of academia, you probably don't benefit as much of it, because we had a lot of projects and topics that we were working on that in the MBA program that required real life experience, and everybody who was in that program had at least 10 years post-academic experience, either in industries or in consulting companies or what have you. So I hope that answers your question.
Yeah, thank you very much. That's actually the first time I get this question actually answered, and that was very eye-opening. Thanks a lot. And then I have a question from the Q&A from the attendees. So someone is asking, how can a fresh PhD graduate source for capital to start entrepreneurship? That's a very good question, I guess.
Well, if you're an entrepreneur, you come up with a plan and you make it happen, right? So there are various sources of funding. And I mentioned, for example, an organization like the Netherlands America Foundation, who provides scholarships to do certain, to get certain experiences. But I think, so in my case, the MBA was paid for by the organization that I worked for. And when I was CEO of Protein Sciences, I also sponsored a couple of people who I really believed were just great employees to do an MBA. And I think so that's probably what you want to do. You want to find an employer who is willing to invest in you. And I hope I made clear in my presentation that nothing is quick and easy, right? The development of the Lipomax took about 8 years before that product became commercial. Flublok took 20 years, and it is a long-term process, and it requires persistence and not give up. And I see, and I don't mean this as a criticism, but I see a lot of young people thinking that, from day one, they should be manager and they should be CEO, and it just generally doesn't work that way, I guess.
Yeah, indeed. Thank you very much.
You're
So
welcome.
I have, there is another question. So it's more about how did you pass from the detergent company to vaccine? So how did you sell yourself for the other company? How did the recruitment happen?
So I had, when I was doing my graduate work, I had worked with baculovirus, and I must admit that the main reason why this company hired me was not necessarily because of a scientific background. It was more because I had an MBA from Rochester University, and they liked the package, and I just basically joined them originally as director of business development, and then within a year I became chief operating officer, and I basically had to run the the research organization and develop a plan for how we were going to get money and how we were going to develop Flublok, because often products don't fail because they're bad products. They most often fail because you cannot get the money together, you don't have the right team, you don't get your regulatory approval. There's all kind of other reasons.
Yeah. Thank you very much. So There is one last question I would like to ask you. It's about knowing a little bit more about featherweight before we close this session. About the movie, how did that happen?
So it actually happened because, and this is, by the way, important, right? Because, you know, it very often you need other people in your career to help you recognize things and advance things. And in Connecticut, I became a member of a group, a women's group that had about 25 women who were all from different industries, and they all you know, came together. We came together once a year in person, where we would share what had been most impactful during that year. And there was, you know, about 5 years ago, a lady, Donna Collins, who spoke about the fact that Connecticut needed reinvention, because Connecticut was a very prosperous state for a long time. But the problem today is that a lot of people leave Connecticut because it's more attractive to live in states where you don't pay as much state tax and don't pay income tax, etc., which is why Florida and Texas and what have you are so loved and desired at this moment in time in the States. And she presented then the life of this boxer called Willie Pep, who basically started working. He was the fighter, the lightweight fighter who won most boxing matches in history. He was very successful, but as he says in the movie, because it's a documentary movie, he earned 4 million and he spent 4.5 million. He had many wives, started, you know, drinking, and got into a decline. His career got into a decline. And then when he was 40, he made a comeback, and he actually did 8 more matches, and he did win some of them. And it's really a very interesting documentary about resilience and about, if you really want to do something, you can do it. So, and I know that Martin asked, did you have mentors? Yes, of course I had mentors. And I think one advice that I can give is, it's so important to work with other people, and especially women. Women so much want to just do their job and do their job well. But I learned over the years that networking, building relations with other people, that's really what gets you there. Because when somebody else would speak about Flublok and say that it was a great product, it had way more impact than when I would say it. So, you know, it's just important to identify those people around you who can lift you up and help you with your, you know, with your career.
Indeed. Thank you very much. So I'm looking forward to watch Featherweight on Netflix when it will come out.
Good. All right.
And so I think we are at the end of the webinar. So thank you very much, Manon. That was great. Thank you also for answering the question. It was very, very interesting. And thank you, Alexander, also for your talk. It was really a great talk. So, and thank you to all the attendees to stay here until now. So I would like to remind you again, before we stop, that on the 7th of February, we will have our next webinar. So please participate. It will be again at 4 Central Europe time. So a few weeks before, we will send around the publicity. So don't hesitate. And also don't hesitate to join us in case you're interested in what we're doing. With this, I say goodbye to everyone. Thank you very much to our speakers. Thank you very much, Martin, and have a great evening or day.
Thank you for your moderation. It was really well done. Thanks.
Thank you very much.